It's my pleasure to welcome you all to our first 20th anniversary symposium. This is something we've never done before, so we're pretty excited about it. It's an opportunity. I should be thanking really the organization committee. So Stuart Lindsay, Steve Monk, and I know there's a number of other ones. Josh Hyeth is here as well. All of them have played a big role in organizing this event, and a key theme, as you obviously will have detected, is bringing back some of our colleagues who have initially been at Biodesign early on and who have gone on to other things. And it's our chance to kind of reconnect with them and hear what they've been up to and all the science and interesting things that they've been doing. So you're going to be hearing that throughout the day. So welcome, and I'm hoping that this will be a relaxed event where people will have some conversation, we'll get some good questions going, and get a chance to reconnect and network with each other. I'm gonna, are there any administrative things I need to tell them about? I mean, the bathrooms, everybody knows, are right outside the door here. So, and there will be lunch later, so. Okay, so it's my special pleasure to introduce our first speaker, our kickoff speaker, who has been a friend of mine for many, many, many years. We've been scientific colleagues and friends for a long time. Ordinarily, this would be the moment to tell you a little about his background, but he's going to be telling you about his background as a big part of the talk, so I won't spend a lot of time on that, other than to say that Parag is just an amazing individual. You don't get to meet people like this often. His brain works in countless different directions, so he understands core science. He spends a lot of time thinking about computation and math and programming, and you're going to hear about some incredibly creative science that he's developed that's led to a company that has now gone public and that is about to come out with a tool that will utterly revolutionize proteomics. It's going to change the way we think about studying molecules because of its ability to look at individual molecules and look at their modifications and how they behave. So all of that's going to be, he'll tell you all about that. He's also a magician and an accomplished one, a member of the Magic Castle in LA. So I don't know if you're planning to do any of that today, but you've all seen, many of you have seen that because he came here once before and did a talk on that. But anyway, so I'm going to bring Parag Mallick up here, and he'll tell you all about the rest of this.
Thank you so much, Josh, and thank you, Dr. Lindsay, for inviting me back again. It's really an incredible privilege to be here and to kick off today on such an amazing occasion. I do feel a little strange being the kickoff here today when we had Carolyn Bertozzi here last night, and we have folks like Josh and Dr. Lindsay and Lee Hartwell and others who are just amazing, and their stories are also enchanting. But what I'm excited about is that what I'm sharing with you today is actually a story that I've never told before. I have never shared my history or my biography or how I got here before. And I'm hoping that through sharing it with you all today, that it will help you as you write your own stories. And I think what I've sprinkled in are a few of the lessons that I've learned around the way from the amazing people who've touched my life. And I've also found myself in a place that I certainly never expected to be. And so we'll chat a little bit about that too. As Josh mentioned, I wear a lot of different hats. One of them is that I am a professor at Stanford, and I run a lab that is incredibly interdisciplinary, looking at multi-omics and next-generation machine learning, and also developing new technologies and instruments and toys, and really trying to understand diseases, how they progress, and how we can identify biomarkers to catch them in their earliest stages and predict their behavior and maybe their treatment. In another hat, I as Josh mentioned, have been developing founder of a really amazing company that, again, lives at that intersection of life sciences, computer and data sciences, and physical sciences and engineering, which is so much of the model here at Biodesign of bringing disparate people together. And the goal of that company is to look at the proteome, the proteome being all the proteins in your body or in a system, and the technologies that we have today are unfortunately a little limited, and we'll chat about that too. And then I do have this third life. Oh, and now I have a microphone that is much louder. Where as it turns out, I am also a professional magician and circus performer, which is something that sounds like it should go well, that magic and science don't complement each other, but as it turns out, they really do. And part of why I am putting this up front and center is because this was something that I hid for a long time. I lived two completely parallel and distinct lives. I did not tell my science colleagues about this for fear that they would look at me and be like, oh, magic? What's that? He can't be a serious scientist. And I would like to disabuse you of that notion that you can only be one thing. And the last time I was here, I did, in fact, give a very different talk. Which Josh was kind enough to be part of. He was sort of voluntold, I suppose, about the convergence of science and magic. And really, as I was developing this lecture today and going back in history, what became very clear to me as part of that were a couple things. One, that parts of my story seem like a trashy novel. And if I hadn't actually lived them, I would be very curious about where to buy this trashy novel. And the other was that a lot of the moments that I thought were the most critical, the most important in my life, weren't. In fact, they were all preceded by other moments that really set the stage for them occurring, that I, at the time, maybe didn't appreciate and maybe didn't notice. And so these seem like momentous occasions: getting your PhD, getting married, starting a company and taking it public, doing a magic show for 5,000 people. But really, this is just the final step in journeys that began chatting with a camp counselor, applying to be part of a nanotech center, buying a truckload of hard drives. I'm serious, this will make sense later. And even taking a linear algebra class. And so as you think about where you are today and the experiences that you're having, pause every now and again and think about how those seemingly intangible moments may actually be the most important moments of your life, that are setting you up for something that you don't know what it is yet. You might not even be able to imagine what it is yet, but it's changing and influencing who you are and how you're going to get to where you want to be. So let's start at the beginning. Where it began is in San Mateo, California. Who here has heard of San Mateo, California? Really? That is shocking. So if we zoom in, there's really usually one reason why people have heard of San Mateo, California. So I grew up right around here. Is that showing up? No, not at all. Okay, what about here? Nope. All right, that's fine. Yes, all right, fantastic. And I went to school right here. Now, the people who are familiar with San Mateo, California usually are because of this other person who lived in San Mateo, who grew up about 2 blocks from me and went to high school literally across across a fence from where I went to elementary school. Anybody know who that was? This person of fame from San Mateo? Steve Jobs? Steve Jobs was not from San Mateo. He was from Cupertino. Close. I'll give you a hint. Does this help? I know we are filled in a room with scientists, and so, however, maybe this helps. San Mateo, California. He grew up 2 blocks from me. What was so amazing about San Mateo, and I'd like you to just picture for a moment, and in fact, close your eyes, and picture yourself as you were when you were younger, maybe 5 or 6 years old. You're tucked in bed, or you're in a pillow fort that you made in your living room, or you're just playing and imagining and dreaming of the future and thinking about who you want to be when you grow up. And think back to that person that you were then, and what are the things that brought you joy? What are the things that got you excited? And on the count of 3, I'm going to ask you to shout those out. All right, 1, 2, 3. That was a wild array of things. I heard space, I heard bugs, I heard toys. Despite being an incredibly painfully shy and introverted person, I was incredibly curious. I asked an absurd number of questions, irritating basically everybody around me. That hasn't changed. I just couldn't help myself. I wanted to know how stuff works. And I asked questions about birds, and I asked questions about dinosaurs, and I asked questions about rainbows and magnets, and lots of questions about rockets and space. But I also did ask harder questions. I asked, why did my dad have to die of cancer? And where is he now? And he died when I was 2.5. And I'm certain that my sister is a cancer surgeon, and I am a cancer researcher, in part because of that. But in addition to asking lots of questions, I also did experiments. At 5 or 6, I set up a lab in my closet. It was not a very large lab. And I had apparatus in my closet. This was my first apparatus. They might look like small rubber dinosaurs. They are not. They are test tubes. And so as it turns out, these dinosaurs have a channel in the middle. I was 5. You work with what you got. And so I would do all sorts of silly little experiments with my dinosaurs, acid-base chemistry that I didn't know what that was. I would try to dissolve chalk. I would try to do random little things, experimenting and being a little scientist in my closet, almost certainly without appropriate protective equipment. And fortunately, my mom was incredibly supportive of this, and so ultimately at a thrift store one day, she found this great chemistry set so that I could be a little bit more of an actual scientist. And then, strangely and coincidentally, around the exact same time that I got this, she also found this. This probably doesn't look like anything recognizable to all of you, but this was my first magic trick. And so the way it worked is that you put a coin in, you take the slide, you put it into the little box, you shake it around, you pull it out, and the coin has vanished. It was amazing. I was 6. But my perspective on what things were was a little warped because I thought that chemistry was the study of blowing things up. Now, this is not entirely inaccurate, but it's only one branch of chemistry, particularly involving large nitrogen-containing compounds. But one really important piece of my journey lived next door. I called him Grandpa. He was not. His name was Clifford Sullivan, and he is among the most amazing mentors I've ever had. He left high school after his sophomore year to work in a lumber yard, eventually becoming a mechanic, going to the Boeing School of Aeronautics, and training as an airplane mechanic to work at United. And despite never finishing high school, he was intensely curious about the world. I learned so much about physics and chemistry and natural forces and carpentry from him, starting early in my journey. And frankly, I have met more than a few academics who are a little uppity about their higher education and a little dismissive of folks who maybe don't have it. Grandpa put that to bed pretty early. He gave me a perspective that not everyone is given the same opportunities. That doesn't make them less smart. That doesn't make them less capable. It just means they took a different path, and they still have a lot to teach you. So I also was fortunate to have another mentor. That was my sister, coming back to San Mateo. So my sister is older than me by quite a bit, and so this is San Mateo, my house, Tom Brady's house, and so one of the other places that I was raised was the San Mateo Public Library, because she was working there, and I got babysat there and read an excessively large number of books, because when you're trapped in a library and you're 7, what are you supposed to do with yourself? You can't run around screaming. But also, when she came back from college, she got a job in Foster City, working for this small company called Applied Biosystems that was working on a little instrument, the automated DNA sequencer. And so she was employee 34 of Applied Biosystems in their sequencing division, and I was babysat there. Again, irritating all the scientists, and her making me run sequencing gels before, so I could pipette probably before I could complete coherent sentences. And this was an incredibly important experience because it gave me a perspective on what a scientist was. So, and I'll just point out that Nautilus, my new company, is down here at the bottom. So I haven't actually progressed terribly far from where I grew up. Although we almost moved into a building that was literally right here, where I could have gone home for lunch with my mom every day. But if you go back to that point and you think back to your 5 or 6-year-old self, and you think about what you thought your trajectory would be, this is my elementary school self view of what my trajectory was going to be. I was either going to be a gas station attendant, or I was going to be an astronaut. I really liked cleaning windows. It's really fun. And of course, even in elementary school, you have jobs. So my key jobs were stay out of trouble, stay out of the way, do well in school, and then I also picked up a side hustle stuffing envelopes. So that was my first actual job. As you get older, you move on. So in high school, I had perhaps the most pivotal moment of my life. Given this career trajectory, you might think that I might do one of 2 things for summer camp. I might either go train with Chevron or go to space camp. And so this is me from space camp. We were in the middle of our extended duration mission. We were having a slight raw sewage problem. And this was the summer that I learned about the protein folding problem. I was a sophomore in high school, and this sounds utterly ridiculous, but one of our counselors was talking to us about the challenges of extended duration spaceflight. And there are a number of them, but one of the largest one is loss of muscle mass and bone density. That as astronauts are in space for an extended period of time, their bones weaken, their muscles weaken and atrophy. And to such extremes that one of the first cosmonauts, Yuri Gagarin, who was floating around for a year, came down and then promptly broke both his legs from the atrophy. And he explained to me this thing called the calcium cycle, which involves 2 proteins, protein hormones, PTH and calcitonin, and their interplay with how they keep calcium in your bones, prevent osteoporotic distress, et cetera. And what he said when sort of talking about this, he's like, well, I think I might have asked a question, well, like, why don't we just give people more calcitonin? It's like, oh, well, there are lots of side effects to things, and you can't just, hit the pathway as hard as you want. But hey, maybe if we could solve this thing called the protein folding problem, which is this problem of given a series of amino acid sequences, can I predict what the structures are? We could design entirely new proteins that are able to act more specifically. And maybe we could then use them to alter the regulation of the calcium cycle to help avoid the catastrophic events associated with long-term spaceflight. Now, I am so curious who in the world this camp counselor was, because this was the most prescient and sage perspective that one does not expect to get at summer camp. And yet, today, with things like AlphaFold and the recent Nobel Prize that was given, we are predicting structures and designing structures, and that is the way that the world is. And this was the path that I was set on as a sophomore in high school. Now, this was a strange summer for me because this also was a summer that I spent with my sister in her lab. She was in graduate school in New York, and I went to see my first Broadway show and was delighted. And so on one half of me, my science brain, I was like, oh my gosh, this is great. I need to work on the protein folding problem. And then the other half of my brain was like, ooh, theater's fun. So if you were to ask me at the end of high school what my expected trajectory was, it had a little more definition. Uh, but nonetheless. And so, my expectations have definitely evolved over time. Now, in high school, I did also have a number of jobs. I was a chess tutor. I also worked at the county fair, both hauling things around and in the floral arts division. I worked as an office temp and could type an absurd number of words per minute. But this set me up for college. And I'm not going to go through too many events in college, but I'll give you a quick flyover of some of them. So I started in college, in the college at Washington University in St. Louis, in the College of Arts and Sciences. And I was like, I want to be a bioengineer, because that's a great way to be able to solve the protein folding problem. But I was in the College of Arts and Sciences, and as it turns out, in order to be a bioengineer, you had to be in the School of Engineering. Oops. So I transferred to the School of Engineering on the first day of college, which is amazing that they let me get away with that. Most colleges are not so flexible as to allow you to switch schools the first day of college, but I did. And then I took biology and hated it. I was taught by this amazingly awful professor, who I'm going to name check. His name was Johns Hopkins, literally. And I'm pretty sure he came in drunk every day. But I learned through that process that I hate taxonomy, and I really do like biochemistry and molecular biology, so that was a useful experience. I also took chemistry and loved chemistry. I had the most amazing teacher, John Jean, who just made it fun. It was, I learned so much without realizing I was learning so much. And as a freshman, I signed up with a research lab and had one research mentor, who I'm not going to name check, ask me if I wanted to study DNA or proteins. So basically, my entire life trajectory was set up through a combination of space camp and talking to this one undergraduate research mentor. When I made the decision, he's like, well, people tend to either study DNA or proteins. He's wrong now, but at the time, he wasn't wrong. And I said, proteins, they're the thing that make everything go in your body, so I want to study proteins. So unfortunately, that particular mentor forgot that I existed, despite me being in his lab for a very long time. And so I switched labs halfway through my freshman year because I got irritated that I had been forgotten about. And that ended up being a remarkable thing. Jay Ponder was amazing. Also in college, I then decided that the bioengineering degree that's currently constructed, trying to build an interdisciplinary training program, didn't really have enough engineering or bio in it. It was sort of this watered down major where you came out sort of a bad biologist and a bad engineer, which is not a really great. So I ended up switching majors to computer science, math, and chemistry, and biochemistry through the chemistry department. But the challenge with all of this is that I was now in this weird mishmash of departments trying to cobble together this integrated program in bio and computation at a time when that wasn't a thing. And so I was formally in the computer science department trying to get a computer science degree. And so I worked with Jay Ponder on protein structure prediction, building these pretty sophisticated algorithms. And the CS department was like, that's not computer science. We're not going to accept that thesis. Like, I disagree. However, they're like, your only option is to go do another thesis. So I did another thesis, this time with NASA, and it was in something that they could not dismiss as not being computer science. It was on distributed synchronization of looking at the International Space Station and trying to determine if it was healthy or not. But the pipe that you had to collect information about whether it was healthy or not was very small. You couldn't get a lot of information from it. It was spending most of its time sending down scientific data, not, am I healthy, are life support working, et cetera. And so this, as it turns out, was a distributed database synchronization problem. We came up with a solution called a virtual time machine that involved sampling small packets of information about the health of a variety of systems scattered throughout, having simulation models on the ground of what systems that are functioning look like, and then doing comparisons between them and sending back these checkpoints between models and observations. Does this sound at all like biomarkers to anybody? That's what we do. We have a really incomplete sensor, and we're trying to figure out what's going on throughout the whole body through a handful of snippets of information captured at random times. So oddly, this project in computer science and synchronized databases, which did end up used on the International Space Station, by the way, has fundamentally altered the way that I think about developing biomarkers. So what initially was a really terrible thing, hey, I have to go do a new thesis, turned out to be one of the best experiences and most important experiences of my life. Go fig. And also, again, I was trying to be super interdisciplinary, so I ended up with 7 different advisors because no one could figure out how to train someone as an engineer and a biochemist at the same time. So I had my first advisor because I came into the School of Arts and Sciences. He was an English major who taught me about ethics. I had a computer science advisor. I had 2 research advisors. I had a chemistry advisor. You get the idea. But there were 2 particularly important moments in college. The first was I went to summer school to work on linear algebra and differential equations, and I met this guy, Will Murray. Now, Will, in addition to being a graduate student in math at Berkeley, had a side hustle. He was a juggler. He juggled our textbook. I thought that was awesome. And so I started, I asked him to teach me how to juggle. He introduced me to an entirely new world. And then the next summer, I went to Circus Center and really started to take this a little more seriously. Who would have thought that 20 years later, I would actually be a professional and doing this in front of crowds of thousands of people and at private events for A-list celebrities. And yeah, it all started because of Will Murray teaching me how to juggle and because I randomly took a math class at Berkeley. The other person who was really influential was John Jean. This was the chemistry professor that I mentioned up front. John Jean was an amazing chemist, but what he was also, and more than anything, was an amazing mentor. And so I asked him, hey, I want to go to grad school. I think I want to work in protein structure. Where should I go? That's hard to know as an undergrad. And he wrote down this list. This is the actual list. This is the actual piece of paper. Well, this is a picture of the actual piece of paper. And he wrote down 5 names. And he said, these people are nice and they do great science. And that's important in that order. So I applied to exactly 5 graduate schools to work with one of these 5 people. And this was, again, when I think on my trajectory, if I hadn't had this conversation with him, where would I be today? Don't know. Now, I will say I did also get some bad advice that not from him, but from our school counselor. Originally, my plan involved being MD PhD because that's better for being an astronaut. And then they said, oh, you're too young. You'll, medical schools don't like that. They'll think you're immature. Why don't you go get a master's and burn a couple years and then apply to medical school? And I thought that was just silly. So I just went to grad school and then never looked back. So if we look at my trajectory at this point in time, from elementary school to high school and college, it was a little clearer. I still had a side thought about being a lounge pianist on a cruise ship. I worked in college as a graphic designer and as a software engineer and an office temp and also in lab as a research assistant. And then there was lots of juggling as a hobby. I was not a professional yet. But inkling by the end of college was this thought of, oh, maybe I'll start a new university, because that sounds like a fun thing to do. We don't get new universities very often. How interesting would it be to do that and to lay an entire new foundation? And biodesign is so interesting because you also don't get substantial institutes very often. And it starts with an incredible amount of ideation and thoughtfulness, and it allows you to do something that no one's ever done before. So that's why this was sort of exciting to me. I also had seen my professors being really frustrated, and so I had a little bit of a thought of like, okay, well maybe I'll start a company to commercialize the amazing advances that are going to happen in protein folding and create a companion research institute with the money from that company so that cool scientists could just do science all day and never have to write a grant ever again. This is an incredibly detailed thought that I actually wrote down and mailed in an envelope to myself. So I clearly have not done this yet, but yet. And then ultimately I went to grad school. The flyover version of grad school, just in the interest of time, is that there were some moments there that were tough. Early on, I didn't think I was going to get into Dr. Eisenberg's lab. That was my entire purpose in going to UCLA. And so if I can't get into Dr. Eisenberg's lab, I'm going to bail on grad school and go do something else. Thankfully, I did end up in Dr. Eisenberg's lab, so I stayed in graduate school, and it was amazing. And along the way, I got to be part of some amazing projects, including building a tool to automatically predict the protein structures of every protein in a series of proteomes. 20 years ago. Unfortunately, my tool was not AlphaFold, and so most of the structures were almost certainly wrong. But then one of the most pivotal moments and choices came halfway through graduate school. I got an email from a recruiter, and that recruiter said, hey, there's this startup company, it's in Westwood, which is where UCLA is, and they're making a series of tools to make it possible to view web pages on your telephone, which is actually a pretty interesting challenge. This was on a normal non-smartphone. There are some interesting computer science challenges. You're basically writing a translating web server that's translating every web page that comes in. Ended up saying, you know what, I'm good. I really enjoy what I'm doing. And so I didn't do that. I turned that down. That was a choice. Now, that company did end up getting sold to Nokia and became the basis of their initial pre-smartphone ability to view web pages. I don't know who I would be. But part of my thinking was, this is important to me. Working in bio and doing something impactful in health is important to me. And I don't know that this is going to help me on that journey. And I don't know who I'm going to be if I take this path. So maybe I'm enjoying the path I'm on. Let me do that. Now, there was one other important thing that occurred in graduate school, which had to do with, so this is Dr. Eisenberg, by the way, in the bottom right-hand corner. About 3 years in, I had a boatload of papers. By all the traditional metrics in the lab, I was ready to graduate. And I had the conversation with Dr. Eisenberg. I was like, all right, I'm good to go, right? Like, I've got all the papers, I've got all the things. And he's like, no. What I want you to do, so you know that rotation project that you worked on a couple of years ago that you then promptly forgot about and moved on to other shinier objects? I would like you to go back and finish that project. And this project involved looking at protein structures, understanding the environments that individual amino acids were in protein structures, and then building some really sophisticated views of what those environments are. And I had tried this project multiple times, failed miserably, and that's why he wanted me to work on it. It ultimately did get published. And the lesson that I learned from that experience of pushing through that project that I was pretty sure I couldn't do and wasn't even sure if it could be done, this project itself was not hugely important. This experience was. This experience that gave me the confidence to try hard things, to not give up on hard things, and to maybe think that maybe I could do a little bit more than I thought I could. And that's the lesson he wanted me to learn. It wasn't that he cared so much about this specific project. What he cared about was that my graduate experience give me a foundation for the rest of my life. Now, one of the other things that I couldn't have seen at the time was that I also got to do a different project that was the most fun I'd ever had. There is this dogma about what are called disulfide bonds. Disulfide bonds are these little connections inside of proteins between two adjacent amino acids called cysteines that help hold the protein structurally together. And there was some very early work. If you look in your classic biochemistry book, it'll say, okay, these only exist outside the cell. Inside the cell, the environment is not compatible with having disulfide bonds. So they don't exist intracellularly. But we were looking at a hyperthermophilic archaea. And we had a crystal structure that showed, of an intracellular protein, that showed two disulfide bonds. So the dogma, the textbook, literally said this cannot happen. This does not happen. But we had a crystal structure that showed it. So maybe the textbook's wrong? Does that happen? Are textbooks wrong? Yeah, textbooks are wrong. I didn't really appreciate that. And so we did this giant structure prediction at the time, again, literally predicting the protein structures of every protein in every bacterial and archaeal genome that was available at the time. and generated this map of where we saw in those predicted structures that were two cysteines that were really close together and could potentially form disulfide bonds. And you can see some of these predicted structures right here, and you can see the little places where the cysteines might be able to couple together on those predicted structures. And what we found was that there were a whole class of organisms, particularly ones that lived in really hot environments. And by I say really hot, I mean like 103 degrees Celsius, that all seemed to have potentially a large number of disulfide bonds, things that shouldn't exist. And so for the first time in my life, I felt like I had learned something that no one had in the world knew before. Like I'd done science, really, and it was thrilling. But I also want to emphasize that at the same time that this was going on, there was something else going on. Also in Los Angeles is this place called the Magic Castle. It is a magician's guild, and it feels a little like Hogwarts. And at the same time that all of this amazing science was going on, I was taking magic lessons from first Mark Wilson, who was one of the most famous magicians, the first television magician, learning the craft of magic and becoming a little more serious about it. You don't have to do one thing. You can do multiples. It's okay. And this was amazing. It complemented my science perfectly. Now, I'm gonna cruise through my postdoc, but I'd just like to highlight one further point along this theme, which is my postdoctoral mentor, Rudy Ebersold. I learned a tremendous number of lessons from him. These are my lessons from Rudy, which involve putting biology in the driver's seat, trying new things, being unselfish, sharing your science with the world, sharing your tools with the world, the value of data and the value of collaboration, as well as having fun and enjoying your life. Now, Rudy was, as part of my postdoc, was sending me to Europe to collect a data set. And I made a deal with him. It's like, okay, I'm not gonna take a break between grad school and postdoc, but when I'm over in Europe, can I take some time off then? And he was like, sure, that sounds great. And so then serendipity comes in at this point. So there I am in Europe, and after collecting this data set, I'm like, all right, I'm gonna go meander around a little bit. Don't know how long, just a little bit. And I'm standing in a park in Salzburg and juggling, as one does in parks in Salzburg. And this person comes up to me and starts speaking in Spanish, which conveniently, growing up in California, I actually knew. And that led to several months of wandering around, literally running away and joining the circus. In the form of a group of traveling street performers. So we didn't have a big top, but it was an amazing experience in the middle of my postdoc. And I have also found, and I'm happy to tell you about that time, it was amazing. But I also learned a lot about people, and I learned a lot about life. And then I also learned that when I was struggling with really hard problems later, I would go to Gasworks Park here in Seattle and juggle. And that sort of zen allowed me to think a little bit more creatively and a little bit more differently. And so the juggling and the magic, again, complementing the science. So my trajectory over time at this point, hadn't changed a whole lot, except that when I started my postdoc, and by the end of my postdoc, I'd picked up this other life. So I had this parallel life as a semi-professional magician and circus performer that I kind of wanted to continue. But like I said, I didn't really tell anyone about it. I also didn't tell the circus performers I was a scientist because I was afraid they would look down on me. I did pick up a third life as building houses as well, which was great, and maintained my my other side projects. Okay, so my first faculty job, I'm just going to give you a teaser. My follow-up interview for this job, usually there's an interview, then there's a chalk talk, and then there's an offer. I had an interview, a chalk talk, a meeting with Steven Spielberg, and then an offer. Okay, that's enough about that. But then one of the most important things in the first part of my first faculty job at UCLA and Cedars, a little bit at USC, was that we generated a crazy amount of data. And I mean, a crazy amount of data at the time. We were generating 1 terabyte a day. And so even today, that's a reasonably good amount of data. It's not a crazy amount of data, but back then that was a crazy amount of data. And so the only solution that we could find was this brand new startup company called Isilon that was making these really fancy hard drives that could handle that kind of volume. They were still, and we were their first beta customer in the life sciences. And their founder was a guy named Sujal Patel, who ultimately became a close friend of mine. And our lab wouldn't have functioned if we hadn't been able to harness this scale of data. Now, at USC, sometimes people continue to give you terrible advice. So my department chair sat me down one day and said, "Oh, I see that you're splitting your energy between bio and technology, computer science. I really think you should just focus on the biology." And I was like, the entire purpose of hiring me is that I do both. I live in the middle. That's why, like, I spent so much effort trying to live in the middle. But this was his perspective. And it wasn't like I was being unsuccessful. Like, again, I had Nature papers, the largest NCI grant with Josh in school history. And so sometimes people give you bad advice. I listened. I didn't follow it, but I listened. My trajectory continued to get even more complicated after my first faculty job, picking up even more jobs. Because again, you don't have to be one thing. Astronaut was still there a little bit, as well as the company institute thing. I have, incidentally, just for completeness, I am not currently an astronaut, in case you hadn't noticed. I have, however, applied to be an astronaut and did once make it in the top 200, which out of 20,000 applications isn't bad. So after this experience at USC, I got an opportunity to go home. There was a new institute being created at Stanford. I was so excited that to be part of building a new institute, building a new center, going home to be closer to my family. And this could have been the most exciting and wonderful moment in my life. It wasn't. So literally my first day at Stanford, my department chair sat me down and said, "Hey, I know you were at UCLA and USC before, but this is Stanford. And if you want to make it here, if you want to be successful, you need to be in the building at least 355 days a year." It's not like he was saying you could have, 10 days off. He was saying you could have 10 days off. And also all those hobbies, professions, make them go away. This advice nearly destroyed my career. I was miserable. I spent 3 years trying to be him and trying to do science his way. I submitted 100 grants and had 0 funded. Again, despite my previous job where I was well-funded and well-published and doing great. And now I was at this premier institution and tanking. And so I started looking around for what else a guy could do. And I got approached by a recruiter. And they said, "Hey, there's this company, Hampton Creek, that is making fake mayonnaise. And they're trying to make fake eggs that scramble well," which, as it turns out, is a really interesting synthetic biology challenge. And as a systems biologist, this is a fun thing to think about. And it was funded by this guy, Vinod Khosla. And that's mostly important because it helped me learn more and more about the world of startups and the world of venture. I didn't take this job. I thought about it. I thought about it really hard. But my lesson there was that it's not about avoiding the challenges. It's about getting back up. And so at that point, after doing this series of interviews, I also interviewed with Impossible Meat. I interviewed with a bunch of other places. I stopped trying to be my department chair. And I was like, this isn't working. If I'm going to fail this way, I might as well fail my way. Let's try something different. So I got back to juggling. I created a new magic show, which is what I presented here several years ago. on what scientists can learn from magicians. And I stopped being embarrassed about being a magician and a scientist to both communities, and was just like, look, I'm both, deal with it. And if you don't like it, it's your problem. Owning it, finally becoming an integrated person for the first time in my life. And then I stopped writing grants just to write grants that I thought were fundable. I wrote things that I thought were fun. I focused on cool science that I thought was interesting, that I actually wanted to do, not because it was strategic. And I also had an amazing experience with a student who was a first-generation student. He initially dropped out after high school. His mom and his dad was in jail. His mom never went to college. And it changed my whole perspective on what I could do as a professor. That these folks who maybe are not, it reminded me of grandpa in a way. Sometimes folks don't have opportunities. That doesn't mean they don't have potential. And it changed the way that I think about building my lab. So I changed the structure of how I choose students and how I bring people into my lab. And I also allowed myself to leave the building and have a life. And shockingly, despite doing all of these things, 6 months later, I was fulfilled. I enjoyed the science that was going on. I lost 20 pounds and was super well funded. And I don't have a great explanation for why, despite entirely changing my personal definition of success away from traditional metrics of papers and grants, I ended up having success by those metrics too. I don't have a good explanation for that. It was a nice consequence. But what I was focused on was do cool stuff, help people. And it worked. And so things were basically good, but I was reaching a point of being a bit crunchy. I was continuing to do magic. And in case you would like a concrete example of what magic can do and how it connects with science and perspective, I'd like you all to please read this sentence for me, or phrase. Come on, what does this say? So as a magician, I would tell you that your brain is constantly lying to you, and that you have lots of cognitive biases, and that magicians exploit that. I would also tell you that magicians live in this crazy world where they are exploring these fine lines between the impossible, the seemingly impossible, the improbable, and the unexplained. And that's what we do as scientists too. In fact, we take things that maybe seem impossible and make them possible every day. So at this time, I was also thinking a lot about proteomics, and I was frustrated. It's like, well, proteins are the drivers of biology. And yet, if you look at the number of publications in genomics versus proteomics, it's a huge difference. And what this came down to was technology in part. It came down to a problem that the tools that we had at the time, if we look at a map of the proteome and sort of how abundant different proteins are, and we look at what was accessible by the tools that we had, the mass spectrometry tools and others, it's this little red insert. We do better in cells, but still, there's a gap. And then there's this whole other axis of proteoforms, which are the multiply modified forms that were just completely inaccessible. And so in the back of my head was rumbling this question of, all right, if you were to start over from scratch and build an entirely new platform, what would you want it to do? And so made a list and said you want it to measure everything, you want it to be super sensitive, you want it to have a range to capture everything, and be reproducible and robust, and also easy to use. Part of the challenge is that genome sequencers are pretty easy to use. Mass spectrometers are not. They're incredibly powerful, amazing instruments that are not easy to use. And so if we want proteomics to be in the hands of every biologist in the world, it needs to be easy to use. So this was rumbling around in my head, alongside being crunchy. So what did I do? I took a road trip. This is Spooky. He was my road trip companion. And I wanted to do that like epic road trip that everybody hears about, where you like drive from coast to coast, and it was too expensive to do a one-way car rental. So I wrote a program to calculate the longest, cheapest one-way rental, which turned out to be from Detroit to Denver. So that was the entirety of my plan, was that I I was like, all right, I know I have to be in Denver in a couple weeks, but every morning I would wake up and pick a direction and go there, which was the exact opposite of my normal life, which is overscheduled to death. And along the way, I did all sorts of random shenanigans. I stopped off in Detroit and joined a random group of people doing acro around the city, winning a prize. I went to see random circuses. If you haven't been to Baraboo, Wisconsin, I highly recommend it. They have the largest collection of circus wagons in the world. At the Ringling Brothers Museum. I also worked on a farm for a second with an old friend of mine, my scoutmaster. And I saw beautiful things and I saw strange things. This is Carhenge, by the way. Why there exists a Carhenge in the United States, I'm not sure. And then I got back, and the weekend after I got back, I woke up on Saturday morning, and my wife remembers this clearly, because I sat up in bed, and the first words out of my mouth were, "Oh, that's how you do it." Promptly followed by, "No, that can't possibly work." And then a frenzied weekend of just utter insanity, of doing simulations and muttering and talking to myself, and things I don't tend to do. I tend to be a relatively restrained human being. And that led to what at the time was called Enigma Biosciences, which is a terrible name for a company. Don't call your name Enigma Biosciences. Venture capital firms hate that. They don't want things to be unknown. And the thesis of the company was, well, right now in proteomics, and again, this was many years ago, mass spec has gotten substantially better. The tools that we had were slow and expensive and could only measure about 8% of the proteome. And with this approach that we were envisioning, that I was envisioning, it seemed like it might be possible to measure 98% of the proteins in a sample and do it quickly and relatively inexpensively. And once you see that, you don't have a choice. You have to figure out how to bring that into the world. So the sort of one slide summary of what we were trying to do was, if you look at genome sequencing, it's pretty amazing. Like, you get most of the genome, you do it completely and easily and straightforwardly. And that wasn't true of proteomics, and could we introduce an approach? And the approach, and so I should mention this slide and this other one, these were from my very first attempt at building a pitch deck. And so this was too. And so the idea behind the method was that we would first take every protein from the sample, and we'd spread them out on a surface such that each molecule lived in its own coordinate. Not type of protein, but molecule. We would then, one molecule at a time, probe them over and over and over again with different affinity reagents that recognized very short amino acid segments, 3 amino acids long. So these are not reagents that recognize individual proteins, they recognize little fragments. But by doing this over and over and over again, you could take that information and put it into a machine learning algorithm and infer the identity of the protein by saying, oh, you have this epitope and this epitope and this epitope and this epitope. So that was the thought. All right, so now you've got a thing. How do you get it into the world? You have options. You can do it in your lab. You can potentially talk to friends who have big companies and be like, hey, you do it. Or you can start a startup company. And after a lot of thinking, it was clear that the best route, this was an idea that required parallelism and scale and collaboration and interdisciplinariness, and that was really mostly compatible with a company. And giant companies aren't great at building new things. And so a startup was the right way to do it. So step 2 was getting some IP. So this was literally when I went and talked to an IP attorney in the Valley, he said, wow, that's the first new idea I've heard in a long time. We need to file on that tomorrow. So his associates, these guys, were up all night writing the first provisional for this idea, which got filed the next day. And so this is it getting filed. And we've since had 8 allowances from that very first patent application. So 8 patents have been issued from that. And then you go and you get some advice. So I reached out to my buddy Sujal, who I'd bought a lot of hard drives from before. And this is literally the email I sent him. hey, we want to buy some storage. He, at this point, I should mention, had sold Isilon to Dell EMC for about $3 billion and was sort of retired, as one does when one sells a company for $3 billion. And was pretty bored, frankly. And then I also, any chance you have time to chat, I could use some advice, I think I have to start a company. And Sujal and I sat down. He called me up and he's like, tell me about this thing. We sat at a whiteboard for about 2 days. And he said, well, I'm kind of bored. I'd love to join you on this. And I'll write the first check. And so that's how we got started. I got a co-founder who actually knew how to run a company, a first check, and we were off to the races. So the very first steps were we rented exactly one lab bench and exactly one desk at a place called StartX, which is around the corner from Stanford. And we were interestingly sitting right next to Tyler Schultz, who was the Theranos whistleblower. I was also on his graduate committee, which was funny. And then today, the platform's evolved a bit. And so I will show you in the fancy video, if I can figure out how to make it actually go, how the platform looks a little bit more today. So we now do exactly what we started, except it's now a very detailed pattern array where we glue down each individual molecule. We introduce these fluorescently labeled affinity reagents that recognize these very short epitopes, rinse off the ones that are unbound, and we take a picture. And so there's no mass spectrometers, they're just pictures of whether there was a light up or not. And we take series after series of these, building up increasingly more information on a per molecule basis, which is kind of amazing. And as a biochemist, what you're looking at here are literally pictures of individual protein molecules. That's pretty freaking amazing. I'm astonished every day that we are looking at individual protein molecules. And then you take that information and you put it into a machine learning algorithm. And so I'm getting close, Josh, so probably 5 more minutes. And then ask the question, okay, here's the pattern of epitopes that I observed. What protein must be compatible with this? And once you've identified every molecule, you just count them up, and that's how you get your quantity. This is what we're building. We can't quite do this yet, but we're getting close. So, the timeline of this started in 2016 with this crazy weekend. And then our first bench, and then our seed round, and then ultimately a Series A. This was our very first flow cell that I used to be the manual liquid handling robot, our first patterned arrays, and on and on. And, but not everything was this smooth, simple journey. One of the sort of hardest things occurred that we were in the middle of our Series B in March of 2020. Can you all think of anything that might have happened in March of 2020? So literally, we're on the verge of closing our round, of getting our Series B investment, when we got a call from the venture partner at this firm. And it turned out that one of the first cases of COVID in the entire United States was in their partnership and had exposed their entire investor base to COVID. Yeah, oops. And so they stopped all investment for 3 months because they were so freaked out. And you can see we had a number of these different planned visits and evaluations, postponed, rescheduled, postponed, canceled. Literally our very first partner pitch, which is where you're supposed to get the entire partnership together and talk about the company, was March 16th. It was the very first Zoom partner pitch they had ever done. The stock market lost 40% of its value that day. They were not paying attention to us. They were sitting there watching their phones and watching the stock market collapse. We ended up, it was miserable. We went from being a darling that everyone was really excited about to basically it being nearly impossible to fundraise. Ultimately, we met with about 150 different VC firms and were able to get through it. But it was it was a lift. But again, that lesson that I had learned earlier about it's not avoiding the challenge, it's about getting back up and fighting through them was critical. Now, one other just quick anecdote from here, which is sort of fun, is that over time we grew and we ended up going public, which was amazing. When you go public on the NASDAQ, they post a little thank you on Times Square. And so this was the post of us listing us. And then they also give you the opportunity to have pictures of influential people in your company up on Times Square. You might recognize this one. Now, and most people, for their photos, they are wise enough to go with a photo that shows them in a very good light. And certainly our marketing department made me take a whole bunch of those. But I snuck one in. So it's been an amazing adventure. And one of the things that I'm so excited about right now is that just a couple weeks ago, we shared the first real biological application of the platform with the world. And I'm not going to go into detail about this terribly much, but this was really the second time in my life where I felt that I really learned something, because we were able to measure proteins with a resolution and sensitivity that had never been done in the history of the world, which is a very grand statement, but there simply wasn't a technology to do it. And we were able to look and say, hey, there are quadruple phosphorylations at these sites on this protein molecule. And this is how many there are. And this is how it changes in neural cells when you drive them towards Alzheimer's disease. Now, very preliminary study. We need to do this 1,000 more times. But the fact that we were able to do the measurement was just awesome. So, all right, I'm getting close to the end, but I'd like to come back to where I started, because I think I've touched on almost all of these. I think talking about Space Camp and my chat with my counselor, the truckload of hard drives, hopefully you now appreciate that my co-founder and partner in all of this adventure, that's where that came from, that led to Nautilus really getting off the ground in the first place. My life as a juggler coming from Will. But the one story I didn't tell you was about applying for an NCI Nanotech Center grants and that leading to me getting married. So when I was still at USC, before I'd moved to Stanford, I had a collaboration with Stanford. We wrote a grant for a nanotech center of excellence. And every quarter, we'd come into this room. This is in Clark Center in the BioX. And I used to sit in this seat right here in the back of the room. And Christina was an instructor at Stanford at the time. And she used to sit over here, like a good kid in the front of the class. And we didn't know each other super well, but that was the first time we met. And then when I moved to Stanford, she knew me from back then, and she was thinking about faculty jobs and wanted to chat about the process and all of that, transitioning from instructor to faculty. And so she ran into me in the just around the hallway and was like, oh, hey, can we sit down, and I'd love to pick your brain and get some advice. And I promise you this was not a skillful sliming move on my part, but my schedule was so insane that I gave her an option. I said, look, your options are, if you want this to happen soon, we're gonna have to go get dinner. Or, we can meet in my office in like 6 weeks. It's just, my schedule is so packed. And so she said, all right, let's go to dinner. And so we spent the entire time not talking about faculty application processes. It turned out her brother was an amateur magician. She wanted to be an animator when she grew up. And so she also was an artist and a scientist. And so we did get married at Disneyland, or Disney World, just a few years ago. And in the middle of it, we did a small science experiment. You've heard of unity ceremonies. People poured like different types of sand or glass or whatever. And like, you know what? Her research, she's a professor at USC, she studies fluorescence and optics, and I'm part chemist. So I'm like, why don't we do a chemical reaction? Like, what is a better bond than an actual chemical reaction? And so essentially figured out the chemical reaction under the hood of a glow stick, and made it happen live on stage, or live at the front of our wedding, with PPE. Now, I would just like to point out that my apparatus had improved substantially from when I was younger. And the perk of getting married at Disney World is you get to take lots of cool pictures in front of the castle. So some of the lessons that I've learned are, number one, that my expectations of my future are usually wrong. I didn't anticipate Nautilus. I didn't anticipate becoming a magician or a spin instructor. I don't know where tomorrow is going to be, and that's okay. And I continue to pick up new potentials, now thinking about maybe another PhD one of these days. And I'd like to close by just sharing a couple lessons. So these are lessons that others have shared with me that I'm going to share with you. The first is cultivate your uniqueness and own it. Part of what makes you an amazing scientist and makes you special is the way that you look at the world and the way that you look at science. And that comes from the fact that you liked playing with dinosaurs when you were 6, or you went to volleyball camp when you were 12. All of these things shape the gestalt of your experience that allow you to look at your data, to look at your experiments in a way that no one else in the world will. Own it. Another key lesson I learned, and certainly you saw with our Series B, you saw with some of my challenges at Stanford, is that failure is just part of the process. It's not something to be afraid of. As part of being a circus performer, you literally fall on your face 1,000 times, and it hurts because you're falling on your face. But then that 1,001th time you end up and you're standing on your feet and you're like, whoa, what happened? And then you do it again and you fall on your face again. And you keep on doing that, and then eventually you fall on your face way less often. And just recognize that. It's okay. It's even good. And then this last point came from my scoutmaster, who I showed you a picture of on the sod farm. She spent her whole life working on things that brought her joy and fulfillment. She loves working on a sod farm. And what I found in my journey thus far is that by focusing on things that mattered to me, focusing on things that I felt were important, focusing on things that I thought were enjoyable, and defining success my way, not necessarily by grants and papers, but by things that mattered to me, life was better. And then my last bits of perspective, these are just mine. And I leave them with you to percolate on. I often tell my undergrads to not let school get in the way of your education. There's a lot of school that happens outside the classroom. There's a lot of learning to be who you are that happens outside of the classroom. Take those opportunities, those 3 a.m. conversations. Sort of related, while there's certainly things that are wrong, there usually is not a single right. And that's hard because we want to think in these binaries of there's right and there's wrong. But really, there is not a right and a wrong. There are choices. Those choices come with pros and cons, and they have consequences. I could have taken that job as a programmer, writing web pages for cell phones. I would be in a very different place. I did not make a wrong choice. I made a choice. Digging into the why, that's what we do every day in science. But we can apply that lens in our life too. Why is that person cranky? Why are we having a tense conversation? What's going on in their life? Approaching with inquisitiveness instead of judgment. This last point about don't let anyone else define success for you, it's really easy. Every A&P committee tries to do it. They're like, you are successful because you had 3 Nature papers and 4 R1s. No, that is a measure of success. It doesn't have to be yours. And then my last point, which I think is the most important, do you. You will be a better you than anyone else in the world. No one else can do you as well as you can do you. The coda there is when I was trying to be my department chair and do him, I was terrible, awful. I will never be him, and I will not be successful trying to be him. So I'm going to stop there and say thank you. And I know I've run super long. I'm really sorry, Josh.
I don't know if we have questions, maybe 1 or 2 questions? No, we're a little late, but we're good.
Very quick one for Parag, because you said you've got
Hold on, we don't have.
the microphones, right. Very quick one for you. So you fell for chemistry blowing things up. And I'm a physicist, but the funnest course I ever taught was the freshman chemistry, because we blew a lot of things up and did the wonderful thermite reaction and so on. But the fire marshal now makes these things impossible. Is chemistry going to die?
Well, I think that so long as the fire marshal doesn't have influence over what happens in your backyard, it's okay. Yeah.
Any other?
Yeah.
So it sounds like you get into a lot of things and probably have dozens of projects and things of that nature. How do you find following through on those, and how do you find like actually completing those tasks?
So that is also a choice, because there are some folks who really genuinely like just exploring and dabbling in things. And that's great. If that's how they find fulfillment, they should dabble in lots of things. I actually do one new thing at a time and go super deep. But I tend to hold on to it forever. And so while I do try a large number of things, I try to become a deep, serious student of it and an expert, and I, treat it kind of like a job almost. And so that, I think, and that's what I prefer. I prefer that depth. That doesn't make it, again, better or worse. That's just how it works for me.
Thank you.
All right, I think, yeah, we're going to move ahead. Thank you so much. That was awesome.